Hi everyone. In this video, I'm going to start talking about the formation of the neural plate, neural tube, and neural crystals. Okay, so first, remember when we talked about three layers of the embryo, right? So your embryo is now going to officially be three layers. So let's start annotating. Um, so your blue layer, I'm going to label this as your ectodermal cells. Your red layer is going to be your mesodermal cells. And your green layer is going to be your endodermal cells. So remember, each layer, this is you as a baby. You're now three different types of cells. And what we are actually going to be focusing on is specifically this structure right over here, which is your ectoderm. Remember how I like to remember it is ecto is the word for outer. Like, exo almost. Ecto is another word for this. Okay, so let's get more into the details. First, let's remember our ectoderm, right? This picture is your ectoderm. And on some point in the middle of your ectoderm, there's going to underneath your ectoderm actually is a structure located in your mesoderm called the notochord, right? So this notochord, well, all you need to know about it is that it's going to secrete little signaling little molecules that will proliferate, that will basically go into the ectoderm and cause rapid proliferation. What does proliferation mean? Proliferation Proliferation means multiplication. Multiplication, or I guess in this case, multiplication by division, or in other words, you create multiple by cell division. So basically, you're multiplying your population numbers by making cells divide, if that makes sense. Just think of proliferation as increasing the total population number through cell division. That's the best way to put it. And basically, these blue cells over here are going to represent the thickening of your ectoderm. So your ectoderm is going to rapidly divide itself over and over and over again so that that one portion begins to get thicker and thicker. Meanwhile, your notochord will also pull these cells inwards. And what it's going to do is it's going to pull it inwards and inwards and inwards until it joins together, and it's going to kind of create this tube-like structure, which is known as your neural tube that you can see over here. So now let's take a few steps back. What is this purple piece of cells over here? Like, what is that? So basically, when your ectodermal cells, so your neuroectodermal cells are forming, these purple cells will mark the border of your neural plate, right? So it's going to differentiate the normal ectoderm cells from your neuroectodermal cells, or the thickened kind of proliferated portion. And these cells, they don't really play a role per se in the formation of the neural tube, but they play a very, very big role in the remainder of development, right? So the reason is basically these cells, once they form your neural crest that kind of sits above the neural tube. So if you notice, like these cells, once the notochord starts pulling the neural groove, your like neural plate, once it starts pulling it down, it forms a neural groove, right? So once the neural groove forms, you can kind of see the neural crest cells coming together, and then they start joining. And then once the neural tube is completely formed, the neural crest kind of sits on top of there. Once this happens, what the neural crest cells are going to do is they are going to migrate everywhere. And basically, we use this acronym, Motel CASA, to help us understand where all that migrates. So first you have M, right? So your M stands for melanocytes of the skin, right? So this gives you pigments, um, odontocytes, which are the precursor for your teeth, tracheal cartilage. So basically, to keep your trachea open, there's going to be rings of cartilage around your trachea that is derived from neural crest cells. Enterochromaffin cells. So this is the neurons in your intestines. So if you guys remember in the previous video, I talked about how the intestines kind of propel food constantly forward. The constant pulling motion is derived from this own nervous system cells, which are derived from neural crest cells. You have your laryngeal cartilage, which is basically the piece of cartilage that holds your two vocal cords in place. Your craniofacial features, so basically your nose, your facial features are derived from your neural crest cells. Your spiral ganglia, basically when your aorta and your pulmonary vein, I believe, is forming, they kind of rotate each other, and the structure that separates the two of them is called the spiral ganglia. That is derived from the neural crest cells. And then your spinal, your spinal ganglia, which is basically like your, what do you call it? Basically, when you're at the ends of your spinal cord where your spinal nerves begin to leave, there's a little bit of a bulge, which is called a ganglion, and that is derived from neural crest cells. And all ganglias and all nerves are usually derived from neural crest cells. And I also forgot to mention that your pia mater, dura mater, and your arachnoid mater, which are the meninges or the protective tissue on the brain, are also derived from neural crest cells. So the reason I tell you guys all of this is that neural crest cells play such an important role in development. So just imagine if your neural crest cells didn't migrate properly, then maybe you wouldn't have pigment in your skin, or maybe your teeth wouldn't form properly, or maybe your windpipe would collapse, or your heart would grow with two fused vessels, or maybe your aorta is flipped backwards from your pulmonary vein, which would be very problematic. Or maybe your spinal nerves don't form properly, which could potentially result in paralysis, right? Because your spinal cord is responsible for transmitting information away from your brain. If it can't do that, or if the ganglion doesn't form properly, you could end up with incorrect communication, which would result in paralysis, right? And all of these things just goes to show how important and how finely tuned this process is. So just it gives you a little bit of perspective into how much could go wrong in this whole process, right? So let's go over a quick recap of this. First, what happens is in your ectoderm, a small portion of your ectoderm will differentiate be due to signaling molecules released from the mesoderm, and they will become and specialize to become neuroectodermal cells. Okay? Neuroectodermal cells. These neuroectodermal cells will be marked by a boundary called the neural crest cells, which play a crucial role in the development of the whole body, right? Then, as the neurons begin to proliferate more and more and more, as these neuroectodermal cells continue to proliferate, they begin to push inwards, forming a structure called the neural groove. As you can see, the neural crest cells are still present and will work and move inwards to join. After they have joined together, the neural crest cells will begin to form on top a little mountain on top of the neural tube, which joins together on, I believe, day 25 to day 27. The neural crest cells will begin to pile on top, and on the last day, the neural crest will hover over the neural tube, and as time progresses, these cells will migrate to different regions of the body through a finely tuned process. And if they migrate incorrectly, this could result in a multitude of problems ranging from lack of pigment in the skin all the way to potential paralysis or the malformation of the heart. Okay, so now that we have a summary of all of that, let's go into the next slide, which is the closing of the neural tube, which occurs on day 25 to day 27. Remember, the formation of the neural plate all the way to the formation of the neural tube happens over the course of 9 days. So, and the closing of the neural tube happens on day 25 to day 27. So first, the neural tube cells begin to close, right? So the crest cells begin to pinch from the center. From the middle of the tube, it begins to kind of pinch first. And then once they begin to pinch, the neural tube kind of forms underneath it. So if you go back to this picture, first your neural crest cells will kind of begin to join, and then your neural plate will combine together to form your neural tube. And then finally, your ectoderm will kind of form all over it. Like your ectoderm will kind of encompass it over it, right? So it kind of your ectoderm will form completely over your neural crest cells and then your ectodermal cells. Sorry, your neural tube cells. Sorry. Okay. And another part of this is that your cranial end or your head end will tends to close a little bit faster than the caudal end. So your caudal end is your tail end. And let's think about why this might happen, right? Why would your head end need to be closed faster than your tail end? Well, I'm not sure if this is completely accurate, but the way I've justified it is that it's better to form faster than to form later because it's just easier for you to kind of, if it closes faster, then your brain has a little bit more time to develop and it needs more time to develop, right? It needs more time than your hind end to develop. So that's kind of how I think about it. And remember, throughout this whole process, cell proliferation or cell multiplication, like division, is still happening and is still very present, right? So because of this, once your neural tube is completely closed, it's going to form three distinct bulges: your forebrain, your midbrain, and your hindbrain. So what does this mean? This should seem a little bit familiar to you. So your forebrain is kind of the representation of your cerebral cortex, the topmost layer of your cerebrum. Your midbrain structures are your limbic structures, as well as a few deep brain structures. And your hindbrain would be your cerebellum, as well as your brainstem. So that's it for this video. If you guys have any questions, as always, please comment down below or email me at stemwme@gmail.com. And yeah, thank you so much for watching. Bye bye.